So here in these live classes, in the mastery live classes, we have like categorized it in the form of questions, like the frequently asked long questions and answers and the short ones. Like what are the various ways of enzymes regulation, which you get in 10 marks and all that, influence of the various factors on enzyme activity, then the clinical significance of your diagnostic enzymes, that is the isoenzymes. Then you have the short questions like classification of enzymes, their examples, what are the various types of inhibitors, coenzymes, cofactors, enzyme specificity, KM, isoenzymes is definition, markers, biomarkers like the cardiac markers and other markers, then therapeutic enzymes. So all of this from the unit, like the most frequently asked long question answers, the essay type questions, and the short question and answers, all of them, we've put them all together here for the sake of convenience so that you know that what are our goals that we are planning to cover through this series on enzymes. Okay. Then here at Auricle, we have three different subscription plans. The basic one, which is for 3 months and comes at 15,000. Then you have the standard plan, which is for 6 months and comes at 30,000. And the mentorship plan, which is for 12 months and will cost you 50,000. So in the mentorship plan, we have notes available and one-to-one personalized guidance as well. Then you can get additional 20% off by using my promo code as displayed here on the screen. Then introducing you to the core features of Oracle. We all know that Oracle provides you with live classes. Then you also have recorded classes so that you can access your subjects anytime, anywhere as per your convenience. Then notes and MCQs will also be provided, which are based on the latest CBNE syllabus, so that you're able to judge your knowledge. Then a very unique feature of Oracle is that we provide you with personalized counseling sessions. So anytime when you need any kind of extra support and guidance apart from academic help, you can feel free to book a slot with us. Okay. So that was about a brief introduction to Oracle. Now let's begin with today's class. Okay. Okay. So I hope the screen is So today we'll be talking about enzymes. So we have, so it gives an analogy of, like, you can consider there are, imagine in a ballroom, it is filled with various types of molecules and your party is about to start. But everybody is here and there in groups and the party is not going on so well. Now you have that showstopper for that party. When that person, he or she enters, then what happens? The party takes a kick start. So in your body is given by enzymes. So enzymes are those showstoppers in our body. So the reactions, otherwise, without the presence of enzymes in its absence, what happens? The reactions are taking place at a very slow pace. Okay, but with the help of enzymes, the rate of reaction is enhanced to a great amount of level so that they take place at a faster rate. So this is a very interesting analogy. Okay, so now let's begin with enzymes. So very important enzyme classification. So we're going to talk about the classification of enzymes first. And this is very frequently asked. You might get like write in detail about the classification of enzymes and give suitable examples. Okay. So this here is an important question, that is enzyme classification. So let's begin with enzyme classifications. According to the RUBNB classification, your enzymes has been classified into 6 different classes. They are oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Okay. So these are the 6 different and important classes into which your enzymes are being classified. So beginning with oxidoreductases. So they are defined as an enzyme that is used to transfer electrons or hydrogen atoms. So your oxidoreductases, they catalyze what? They catalyze oxidation reduction reactions. Okay, they catalyze. Okay, that is transfer of an electron or hydrogen atom. Okay, so they are your oxidoreductases, and they are again subclassified into, these are the subclasses. So your oxidoreductases, they are again subclassified into dehydrogenases, oxidases, peroxidases, and oxygenases. Now, what is a dehydrogenase? Your dehydrogenase, it removes hydrogen from a substrate and it is going to donate that hydrogen to another substrate. And usually that acceptor, that hydrogen acceptor is your NADH, okay, a B complex vitamin. And usually the most common one being NADH. Other hydrogen acceptors are your FAD and NADP. Okay, here actually this is for dehydrogenase. Okay, so your dehydrogenases, what are they doing? They remove hydrogen from a substrate and donate it to another substrate. Okay, and usually this another substrate that is going to accept that hydrogen, which is going to act as a hydrogen acceptor. Usually it is a B complex AD. Now here you can see this example here. I've zoomed in. You can see what is happening. This lactate dehydrogenase, this enzyme, it is going to remove, you can see this hydrogen here is being removed and it is being accepted by this NAD to form what? NADH. NADH is formed. Okay. So from one substrate, this is your substrate one. So from this substrate, hydrogen has been removed and it has been accepted by this another substrate, NAD. Now, apart from NAD, you have other acceptors as well. That is your FAD, and it can change into FADH and NADP. They can also act as hydrogen acceptors. So that was about dehydrogenase. Then you have another subclass of oxidoreductase, that is your oxidase. Now, in oxidase, the hydrogen atom acceptor is always what? It is an oxygen, okay? So that is the difference between a dehydrogenase and an oxidase. In dehydrogenase, that acceptor can be anyone apart from oxygen. But in oxidase, that acceptor is oxygen, okay? So hydrogen is accepted by oxygen only. Okay, so that is the difference between dehydrogenase and oxidase. Now your peroxidases, they use H2O2, that is hydrogen peroxide, as an electron acceptor. And your oxygenases, they are able to incorporate oxygen atoms into substrate. Okay, now they can either be monooxygenase or dioxygenase. Mono or dioxygenase. So monooxygenase means it is going to incorporate a single atom of oxygen, and dioxygenase means it is going to incorporate both the atoms of oxygen into the substrate. So that was about oxidoreductases, the first class of enzymes, oxidoreductases. Now we're going to talk about transferases. Okay. So transferases, these are enzymes that are used to transfer any molecule so that the molecular formula is changed. Okay, so what is happening? Transferases, your transferases, they transfer in simple words functional group from one substrate to another. Okay, and in doing so, what will happen? The molecular formula is going to change. So they transfer functional group from one substrate to another. Now, let's have a look at the examples of transferases, like your methyl transferase. As the name suggests, it is going to transfer a methyl. Your kinases, they transfer what? They transfer phosphate groups from ATP. Phosphorylase, they transfer phosphate from inorganic phosphate. And your aminotransferase, they are involved in transamination reaction and they transfer what? Amino groups. If you have a look here at this transamination reaction brought about by ALT, that is alanine transaminase, you can see what is happening. This alanine, the amino group from this alanine is being transferred to alpha ketoglutarate to form glutamate. You see, this amino group from alanine has been transferred here, and what has been formed? Glutamate has been formed. And this alanine itself had formed its corresponding keto acid, that is pyruvic acid. Okay. So your transaminases, that is aminotransferases, ALT, AST, they are all involved in transfer of amino groups. So they come under transferases. All your kinases, they are involved in transfer of phosphate group, like your hexokinase, if you remember. What was hexokinase doing? There was glucose. Glucose was being acted by hexokinase. So this hexokinase was transferring a phosphate group from glucose to form glucose 6 phosphate. So your kinase is transferring a phosphate group from ATP to glucose to form glucose 6 phosphate. Therefore, a kinase is also comes under which class? It comes under transferase. Okay. Now, another example is like your methyl transferases and your phosphorylases. Then moving on to the next class of enzymes, that is your hydrolase. They are defined as enzymes that are used to break the chemical structure and use water. So what is hydrolase is doing? Hydrolases, they are breaking covalent bonds, okay? Carbon, nitrogen bond, carbon, oxygen bond. So these are your covalent bonds. So your hydrolases, they are going to break these covalent bonds with the use of water. That is, they're going to use or add water. So your hydrolases, they break covalent bond by adding water. Your examples being your phosphatase. Okay, it removes the phosphate from a substrate, your phosphatase. Okay, and all digestive enzymes are also example of hydrolases, like your amylase, peptidases, and the primary structure. Basically, where are your covalent bonds present? They are present in the primary structure. Okay, so anything that is going to break the primary structure, it is going to be a hydrolase. Okay, so for example, if you take the example of glucose, now in glucose your macromolecule is glucose, and in glucose what are the bonds present? The covalent bonds. You have the glycosidic bond. So any enzyme which breaks this glycosidic bond, it is going to be an example of hydrolase. So, for example, your amylase, your lactase, okay. Or let's take the example of protein and also like your maltase, etc. Okay, all these. Now, let's take the example of proteins. So, in proteins, what do you have? You have peptide bonds, okay. Now, any enzyme which is going to act on these peptide bonds and break these peptide bonds, they're going to be hydrolase. Like your peptidases, okay, and proteases. Okay, so these are the examples. So that is why it says all digestive enzymes, they are your hydrolases. So now it may be like your trypsin, chymotrypsin, or those involved in carbohydrate metabolism, like lactase, maltase, amylase, etc., and all these. Then your nucleic acid, what do they have? They have phosphodiester bonds. Okay, so nucleases are going to break these phosphodiester bonds. So your nucleases also come under the category of hydrolases. Then your lipids, what do they have? They have ester bonds. Okay, and those ester bonds are broken by lipases and esterases. So your lipases and esterases are also example of hydrolase. Okay, now moving on to liases, the next class. They are defined as enzymes that can make and break covalent bonds, but they do not require water or ATP. So now that is the catch point, that your liases, they break covalent bond without adding water. Now, for example, your aldolase here. See, so aldolate, it is going to cause the splitting. If you remember, it was causing splitting of in glucose, in was acted upon by aldolase to form what? Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, if you remember. Okay, so your aldolase, it is going to break your fructose 1,6 bisphosphate into two different molecules, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. It is going to bring about the splitting, but this breakage does not require water or ATP. The other examples are your decarboxylases, but only simple decarboxylations come under liases. Your oxidative decarboxylation, where NAD is involved, it comes under dehydrogenases. Then you have hydratase, for example, fumarase, from malate to fumarase. What is involved? Your fumarase enzyme is involved, and that requires addition or removal of water. Now moving on to the next class, isomerases. Isomerases, they are identified as enzymes that are used to make different compounds, but the molecular formula do not change. So the molecular formula is not going to change. It is only going to catalyze isomerization reactions. Okay, for example, from glucose 6-phosphate to fructose 6-phosphate. So what is happening? Your glucose is changing into fructose. So the molecular formula both for glucose and fructose is the same, but what is changing? The structure is changing. Okay, then your mutases, for example, from glucose 6-phosphate to glucose 1-phosphate, what is happening? This transfer of phosphate group, but within the molecule, from the sixth position to the first position. So that mutase also comes under isomerase. Then your epimerases, they interconvert epimers. Okay, epimers, we know that they were different in orientation of hydroxyl and hydrogen atom at a single carbon. Okay, so epimerases also comes under isomerase. And then you racemase, they interconvert L and D stereoisomers, so they also come under isomerase enzyme. Now moving on to the next and the last class, that is the They are defined as enzymes that are used to make different compounds by using ATP. What are they doing? They are joining two substrate, okay? And also for this, what do they need? They need ATP. So your synthetases, all your synthetases like glutamine synthetase, here we can have a look at this example. This is your glutamate. You can see ATP is involved, and this another substrate. You have NH4 ammonia, and you can see this is being added here, and what is under ligase. Then your carboxylases, they are also adding a carbon to a substrate. Okay, so the other substrate is your carbon dioxide. So all your carboxylation reactions, they also require ATP, and they might be biotin dependent or independent, depending upon if they require biotin or no. So your carboxylases and your synthetases are example of ligases. So with this, we come to the end of the classification of enzymes. Let's have a look again. So enzyme classification, majorly there are 6 different classes. Having said that, what are those classes? They are oxidoreductases, that is, they are involved in oxidation reduction reactions. They are further classified into subclasses that are dehydrogenase, oxidase, peroxidases, and oxygenases. Then you have transferases. Transferases, they are basically involved in transfer of functional groups from one substrate to another. Now you have the examples like methyl transferases, kinases, phosphorylase, and aminotransferases. Then you have hydrolases. They are involved in breaking of covalent bonds by adding water. And all your digestive enzymes, they come under hydrolases. Then you have lyase. They're involved in breaking of covalent bonds, but without adding water. So they are your lyases. For example, your aldolase synthases, your decarboxylase, and hydratase. Then you have isomerases. Isomerases, they are only catalyzing isomerization reactions. For example, phosphohexoisomerase or your glucose 6-phosphate changing into glucose 1-phosphate by a mutase. They're all example of isomerases. And then finally, we have ligases. Your ligases, they join two substrate, okay? They join two substrates. Okay, and also they require ATP. ATP is needed. And so they join two substrate, make a covalent bond, and ATP is required for this. Your example being carboxylases. For example, your pyruvate carboxylase. And your synthetases, for example, glutamine synthetase. So that was briefly about your classification. Now moving on to the next topic, that is coenzymes. So it says, what are coenzymes in their examples? So you might get a question like this, that define coenzymes with examples. So let's see what are coenzymes. Coenzyme is a substance that enhances the action of an enzyme. Okay. The catalytic activity of enzyme mostly depends on the presence of non-protein compounds called coenzymes. Okay, so we all know that enzymes, they are proteinaceous catalysts, okay, biocatalysts. And what is happening to this protein part? Okay, your enzyme has a protein part, and if there is additional non-protein part which is required for the activity of the enzyme, that is known as coenzyme. Okay, now you have, like, if I categorize enzymes, you can have simple enzymes and complex enzymes. So the simple, they only have protein part, but you also have complex enzymes which have protein part plus non-protein part. Okay. Now, this non-protein part, it can be a cofactor, coenzyme, or a prosthetic group. We will see what all it means. So, a cofactor, if that non-protein part is a metal, then it becomes a cofactor. And coenzyme, if that non-protein part is a small organic molecule, then it is a coenzyme. And mostly these coenzymes, they are derived from your vitamins, like your NAD, which comes from niacin, your FAD, which contains riboflavin. Okay, so that becomes your coenzyme. Now, this coenzyme, it can either be a co-substrate. So now your coenzyme, it can either be a co-substrate or it can be a prosthetic group. So co-substrate means that it is temporarily comes and attaches to the enzyme. It is not permanently attached to the enzyme. The coenzyme that only transiently associate with the enzyme, and then it is going to disassociate in an altered way. So that becomes your co-substrate. And the prosthetic group, it is actually permanently associated with the enzyme and it returns to its original form. So that is known as your prosthetic group. Your example being like your flavoproteins. So that was about coenzymes. Let's have a look at the examples that are given here. That see the first one it says lipoic acid. The others are vitamins, all water soluble vitamins. They act as coenzymes like the B complex vitamin, like I told you, and vitamin C. The only fat soluble vitamin that is going to have a coenzyme role is your vitamin K, D, and FMN. Then an example of your metals acting as coenzyme, that is magnesium. So your kinases, they require magnesium, or phosphorylases, and your carboxylases. Now copper is required by oxidases, cytochrome C oxidase, tyrosinase, ascorbic acid oxidase, amino acid oxidase, lysyl oxidase, cytoplasmic superoxide dismutase, and they require copper. Now, molybdenum, it is required by xanthine oxidase and sulfide oxidase. So, we've seen that what are coenzymes, what are cofactors, prosthetic group. So, I'll repeat this once again. Your proteins, they are forming enzymes. Okay, so your enzymes, they are protein in nature. What is happening? You can have either simple enzymes or complex enzymes. Now, a simple enzyme, it is only going to have the protein part, okay? But your complex enzyme, it is going to have a protein part as well as a non-protein part. Now, this non-protein part is known as a cofactor if it is a small organic molecule, or known as a if it is known as a coenzyme if it is a small organic molecule, or known as a cofactor if it is a metal. Now, again, if that coenzyme is like transiently associating itself with the enzyme, then it is known as a co-substrate. And if it is permanently associated with the enzyme, then it is known as a prosthetic group. So, if you, the protein part and the non-protein part together of the enzyme, they are known as holoenzymes, okay? So there is another term known as holo. Have the protein part and the non-protein part put together, that forms the holoenzyme. So this is your holoenzyme where you see you have what? Epoenzyme, that is the protein part plus the non-enzyme, and this holoenzyme, this is the requirement for the functionality of the enzyme. Okay, for the enzymes to function. So the catalytic activity of the enzyme is going to depend on the presence or absence of this non-protein compound. Okay, so the enzymes will be active only in the holoenzyme state. Now moving on to the various types of enzyme inhibitors. This is a very important topic. That is enzyme inhibition. So the major types of enzyme inhibition, there are 6 major types. That is competitive inhibition, non-competitive inhibition, then you have uncompetitive inhibition, allosteric inhibition, feedback inhibition, and suicidal inhibition. We're going to talk about each of these one by one. So talking about competitive inhibition, here the inhibitor resembles substrate in structure, and inhibitor is going to bind at the active site. So it says that your inhibitor, it is going to be a structural analog. Okay, and it is going to, it is a structural analog of the substrate, and it is also going to bind at the same site as the substrate, that is the active site. So let us have a look at the key features of competitive inhibition. It says Michaelis constant does not depend upon change in enzyme and substrate concentration. So your Michaelis constant, basically it was defining the affinity between a particular enzyme and a substrate pair. Okay, that is what is the affinity of that enzyme for that particular substrate. Now what happens in competitive inhibition? The affinity between enzyme, affinity for both substrate and inhibitor. So affinity decreases, so Km increases. Inhibitor is a analog of substrate. Vmax remains same, but Km increases. So in short, what it is trying to say is that in your competitive inhibitor, the inhibitor and the substrate, first point is the inhibitor and the substrate, they are analog of each other. Second, it is also going to bind at the acid where the substrate is binding. Then the other thing is that it is reversible. Okay, that is if you increase the concentration of the substrate, the inhibition can be overcome. So inhibition can increasing the substrate concentration, more substrate when you're giving, so there will be more chance of binding and the inhibition can be reversed. Now another thing is that here your Km increases and your Vmax remain constant. Now the reason to this, it has also explained that it says that your Km is defining the affinity between the substrate and the enzyme. So what is happening now? You also have the inhibitor and the enzyme is also having affinity for the inhibitor. So what is happening? The affinity between the enzyme and the substrate is decreasing, and therefore because of which the Km is going to increase, but the Vmax is going to remain constant. So that was about competitive inhibition. Moving on to non-competitive inhibition. Here, what is happening? The substrate do not resemble inhibitor. So the inhibitor is not a structural analog of the substrate. The inhibitor binds at another side other than the active side. Okay. So now the difference is, firstly, they are not the inhibitor and the substrate. They are not structural analogs. Second, it is not binding at this active site. Okay. Now Vmax is lowered, but Km is going to remain the same. So why is the Km going to remain the same? For example, let's say this is the enzyme. Okay. This is the enzyme and this is the substrate. So the substrate is binding at the active site and this is the inhibitor. So from this diagram, you can see this here that I have drawn, this inhibitor. Inhibitor is firstly not the structural analog of the substrate, and also it is not binding at the same site, that is the active site, as the substrate. Okay. Now what happens? As long as the inhibitor is there, the substrate is not going to bind, and therefore it is going to bring about inhibition. And this inhibition is mostly irreversible. So the inhibitor can bind with the enzyme substrate complex as well as the free enzyme also, but it does not change the substrate affinity for the enzyme. So the binding of the substrate and the enzyme is in no way changing because the inhibitor is not competing for the same, remains same. Therefore, we say that Km is same. But as long as the inhibitor is there, the reaction is not going to take place. The product is not forming. Therefore, we say Vmax is lowered. Okay. So I hope this explains the reason behind this, that why your Vmax is lowered and Km remains same. That was about non-competitive inhibition. Now moving on to uncompetitive inhibition. In uncompetitive inhibition, what is happening? The inhibitor does not have any affinity for the free enzyme. The inhibitor is going to bind to the enzyme substrate complex, but not to the free enzyme. Okay, so here what is happening? Your Vmax and Km, both of them decrease. Now, an example is your acetylcholine inhibition. This is one example of uncompetitive inhibitor. The another is inhibition of calcinyl alkaline phosphatase by phenylalanine. Okay, so here what it is saying that your inhibitor, it is not having any affinity for the free enzyme. It only binds to what? It binds to enzyme substrate complex. So that is your uncompetitive inhibitor who is binding to the free enzyme as well as to the enzyme substrate complex. Now moving on to feedback inhibition or end product inhibition. Here what is happening? For example, let's see, A is changing into B is changing into C, and C then forms D. And the enzymes that are required are E1, E2 for this step, and E3 for this step. Now, this end product, D that is formed here, it is going to inhibit this enzyme E1. So you see the end product itself inhibits the reaction. This end product D here, it is going to inhibit the first enzyme. And this is known as feedback inhibition or end product inhibition. So feedback inhibition is a natural phenomena occurring in the body. It is normally observed in regulation of enzymes and pathways, and product itself inhibits the reaction. Now moving on to suicide inhibition. What is suicide inhibition? Here in suicide inhibition, what is happening? Your inactive compound is going to bind to the enzyme, and via the enzyme mechanism of the enzyme, it is going to be converted into a more potent form and irreversibly bind to it. For example, your allopurinol, it inhibits xanthine oxidase by a proper mechanism, which is suicidal inhibition. And this suicidal inhibition is unnaturally occurring phenomena. So this is not a naturally occurring phenomena, unlike feedback inhibition, which is naturally occurring. Suicide inhibition is not naturally occurring. And it is also known as suicide inactivation or mechanism based inhibition, and it is an irreversible type of enzyme inhibition. It occurs when an enzyme binds a substrate analog during the normal catalysis reaction. This binding leads to the formation of an irreversible complex through a covalent bond. This enzyme, this complex renders the enzyme permanently inactive. So that is suicide inhibition. Once again, I'll mechanism-based inhibition, okay, or also known as suicide inactivation. Now, what is happening? The original inactive inhibitor, that is the inhibitor, originally it is in its inactive form, okay, and it is also in structural analog. But on binding to the enzyme, what happens? It gets converted into a potent inhibitor by using the enzyme's own reaction mechanism and thus inactivates the enzyme. So initially, what was happening? That the original inhibitor was inactive and it was in structural analog. It gets converted into the more potent form on binding to the enzyme by using the enzyme's own mechanism reaction. Therefore, it is also known as mechanism-based inhibition, okay? And then it newly formed potent inhibitor binds irreversibly to the enzyme. And in the example, you have allopurinol inhibiting xanthine oxidase, okay? So your allopurinol, it binds to xanthine oxidase and it is converted into alloxanthine. And this alloxanthine irreversibly binds to the enzyme xanthine oxidase and inhibits it, okay? Now you have some other examples of suicide inhibition, like 5-fluorouracil acts as a suicide inhibitor or thymidylate synthase. And this, what happens? Your thymine synthesis is inhibited. Now what will happen? Your DNA synthesis will be interrupted. Therefore, this is used as a treatment in fast-growing cancer tumors. Now, another example is aspirin, which inhibits cyclooxygenase. Then you have nerve agents and pesticides like parathion, which irreversibly inhibit acetylcholine esterase. So those were a few examples of suicide inhibition. So recapitulating the various types of inhibitors. So the first one, you have competitive inhibitors. What is happening in competitive inhibitors? The inhibitor is a structural analog of the substrate and it binds to the same site as the substrate. That is, it binds to the active site. Okay. Now what is happening? Your Vmax is going to remain same and Km is going to increase. Why is Km increasing? Because now you have the inhibitor and the enzyme will have some affinity for the inhibitor as well. So the affinity between the substrate and the enzyme is going to decrease. And when the affinity decreases, what happens? Your Km increases. Therefore, your Km is increasing in competitive inhibitors and your Vmax is going to remain same. But this inhibition is reversible, as I've mentioned here. So it can be overcome by increasing the concentration of the substrate. Then having a look at the examples of competitive inhibitor, you have arsenate, you have malonate, you have fluorocitrate, and you have oxalate. Okay. Then talking about non-competitive inhibition, here what is happening? The inhibitor, it is not a structural analog, that is, it does not resemble the substrate, and it is also not binding at the same site as the substrate, that is the active site. It binds to a site other than the active site. But here what is happening? Your Km is going to remain the same and Vmax is lowered. Why is your Km remaining the same? Because your inhibitor is not competing for the same site as the substrate, that is the active site. Okay, it is binding at a different site. Therefore, your Km is going to remain same because the affinity for the enzyme for the substrate is going to remain the same. But your Vmax is lowered. And why is Vmax lowered? Because as long as the substrate is bound to another site, what is not happening? Your reaction is not taking place. And also in non-competitive inhibition, what is happening? The inhibitor can bind to the free enzyme as well as the enzyme substrate complex. And also in your non-competitive inhibitions, they are all irreversible and they're mostly poisons. For example, your iodoacetate inhibiting glyceraldehyde triphosphate dehydrogenase, your fluoride inhibiting enolase, your fluoroacetate inhibiting aconitase. Now moving on to uncompetitive inhibition. In your uncompetitive inhibition, the inhibitor does not have any affinity for the free enzyme. It is only going to bind to the enzyme substrate complex. Okay, and here both Vmax and Km is decreased. Example being your acetylcholine inhibiting placental alkaline phosphatase. Now, moving on to feedback inhibition or end product inhibition. Here, what was happening? For example, I've given you this, that your A was changing into B, then B was forming C, and C was forming D. So, for example, I've just given a random example. Or you can also take like glycolysis, your glucose was undergoing a chain of reactions, a pathway, and ultimately forming pyruvate. Okay. Anyway, so what I'm trying to say here is that the end product that is formed, it is going to inhibit the first enzyme in the pathway. So you see that the end product here, it is inhibiting the first enzyme E1. So this is known as feedback inhibition or end product inhibition, where your final product is going to inhibit the first enzyme. And this is a natural phenomena. Now, moving on to the last type of inhibition, that is suicide inhibition. In suicide inhibition, what was happening? Your original inhibitor, it was not very potent, but it was a structural analog. It is inactive, but on binding to the enzyme, by using the enzyme's own mechanism, it gets converted into a more potent form, and now it irreversibly binds to the enzyme and inactivates the enzyme. The example be your allopurinol, which inhibits xanthine oxidase. Okay, so your allopurinol is going to bind to xanthine oxidase and then it is going to convert into alloxanthine. Now this alloxanthine is potent and it irreversibly binds and inactivates xanthine oxidase. And then apart from that, we have other examples as well, like your 5-fluorouracil. It acts as a suicide inhibitor for thymidate synthase, which is used in cancer treatment, then your aspirin, which inhibits cyclooxygenase. So these are some other examples of suicide inhibition. Now, next we have enzyme regulation. Another important topic, enzyme regulation. Now, the regulation of enzyme activity is very important, and with enzyme activity, it can be regulated by either controlling the quantity or by either controlling the quality, that is the catalytic activity. Okay, so this regulation is brought about in two ways. That is, either you control the amount of enzyme that is synthesized or you control its catalytic activity. So let's see what is the definition of enzyme regulation. Says it is the process by which cells can turn on or turn off or modulate the activities, which are the activities of various metabolic pathways by regulating the activity of enzyme. So if we take the example of enzyme regulation by controlling the quantity, so that is brought about by induction and repression mechanism. Okay, so here what is happening? You are controlling enzyme synthesis, and this is brought about by induction or repression. Here what is happening? Regulation of enzyme synthesis is being controlled at genetic level, at gene level. Now, induction is going to increase enzyme synthesis while repression is going to decrease enzyme synthesis. So, with the help of an inducer or a repressor, what is happening? The structural gene for a particular enzyme, it can be inhibited or stimulated, thus controlling the synthesis of that enzyme. For example, you take an inducer. So that inducer, it is going to stimulate the gene, and in turn, the synthesis is going to be increased. Now, if you take a repressor, the repressor is going to suppress the structural gene, and the synthesis of that enzyme is going to be decreased. So in this way, you are increasing or decreasing the synthesis of enzyme, and thus I say that you control enzyme synthesis. Okay, so that is enzyme regulation by controlling the quantity. Like I said, enzyme regulation can either be like you control the quantity or you control the quality. So, firstly, we're talking about by controlling the quantity. So, that is brought about by induction and repression, which is taking place at the gene level. Okay, and your example being like ALA synthase. If the body is not having enough heme, okay, so the structural genes for ALA synthase will be active, and more ALA synthase will be produced, and in turn leading to more heme synthesis. So that is one example. Other examples include like your insulin, it induces glycolytic enzymes and represses gluconeogenetic enzymes. Or your glucagon, it induces pyruvate carboxylase and represses phosphofructokinase. Or like the third example, like I told you, of ALA synthase. So what is happening? If the body is not having enough heme, the heme level in the body is low. So your ALA synthase, it is going to be active. The genes that are synthesizing ALA synthase, they are going to be active, and more of ALA synthase is going to be produced, and thus more of heme synthesis is going to take place. So that was by controlling the quantity. Now the other way of regulating enzymes is through quality control. Okay, now this quality control, it includes allosteric regulation and covalent modification. So let's talk about allosteric regulation first, and then you see we have covalent modification here. So this is when you are controlling the enzymes via controlling it through its quality or catalytic activity. Okay, now let's see what is allosteric regulation. So allosteric enzymes have allosteric sites different from the active site. So allosteric modifiers, they bind to these sites, either activating or inhibiting the enzyme activity. So what is it trying to say? That apart from the active site, there is another site which is known as the allosteric site. And to these allosteric sites, modifiers are going to bind. Now these modifiers can either be positive modifiers, that is they increase the activity, can be negative modifiers, that is inhibitors and decrease the activity of the enzyme. Okay, now they are classified into K class allosteric enzymes or V class allosteric enzymes based on Km and Vmax values. So what is K class allosteric enzyme? That is, they change Km but not Vmax. Substrate saturation kinetics like competitive inhibition. And your V class allosteric enzymes, what are they? They change Vmax but not Km. So substrate saturation kinetics like non-competitive inhibition. So allosteric enzymes, they also show cooperative binding with positive and negative modifiers affecting active site activity. So what is it trying to say? That what is allosteric regulation? In allosteric regulation, you have those enzymes in which the enzyme activity, the enzyme apart from having the catalytic site is also having a allosteric site. So the allosteric enzyme is an enzyme whose activity in the catalytic site is modified by the presence or absence of a modifier in the catalytic site. So it is trying to say that those enzymes, allosteric enzymes, in which what is happening in the active site, that is the activity of the catalytic site, it is depending upon the presence or absence of a modifier in the allosteric site. Now, such enzymes can only act if the modifier is present. Now, allosteric enzymes, as I said, they have two sites, the catalytic site. I'll write it down. Catalytic site, where the substrate binds. And apart from that, they also have an allosteric site, where what is going to bind? Your modifier is going to bind. Now, this modifier, it can either be an activator, like it says here, see? It can either be an activator or it can be an inhibitor. Now, this modifier can either be an activator or it can be an inhibitor. Okay, so if in the allosteric site a modifier is binding and that due to binding of that modifier, the enzyme is becoming active, then it is known as an activator. And if the modifier that is binding in the allosteric site, again, because of which the enzyme is becoming inactive, then it becomes inhibitor. But depending upon the modifier, you have two types of allosteric regulation, that is allosteric activation and allosteric inhibition. Like I said, if the modifier is an activator or an inhibitor. If it is an activator, it is allosteric activation. If it is an inhibitor, it is allosteric inhibition. So what is happening in allosteric activation? The catalytic site is not favorable for the substrate to bind, okay? But when the modifier is binding to the allosteric site and that modifier is an activator, what happens? That unfavorable catalytic site is converted into a favorable catalytic site for the substrate. Okay, I'll repeat this once again. What is happening? You have an allosteric enzyme. This allosteric enzyme, it is having a catalytic site and it is also having an allosteric site. Now to the allosteric site, what is happening? A modifier or an activator comes and binds. Now, because of the binding of this activator, the unfavorable catalytic site, it is being converted into a favorable catalytic site. So what will happen? Your enzyme is going to get activated. Now the reverse is going to happen in allosteric inhibition. The allosteric inhibition, what happens when the modifier, which happens to be an inhibitor, it comes and binds to the allosteric site. So your favorable catalytic site is going to convert to an unfavorable catalytic site. So that is allosteric inhibition. So that sums up about about allosteric regulation. Now moving on to covalent modification. This is another way how you regulate enzyme activity. That is through covalent modification. Now this covalent modification can either be irreversible covalent modification or it can also be reversible covalent modification. Now what is irreversible covalent modification? In irreversible covalent modification, you have your proenzymes or zymogens. So some enzymes are synthesized in inactive proenzyme forms. Then they are converted into active form by irreversible breakage of specific covalent bonds. So what is happening? You have initially the enzymes, they are being synthesized in the inactive form. Then because of breakage of certain covalent bonds, they are being converted into active forms. So example, your proteases in the GI tract, like your trypsinogen being converted into trypsin, your chymotrypsin, and your trypsin and all that, they are examples of zymogen activation. So significance of this is that they protect tissue from auto digestion and facilitate rapid activity. So that is example of irreversible covalent modification. Now what is reversible covalent modification? This is brought about by addition or removal of a group. Okay. So what is happening? Most important reversible covalent modification, phosphorylation, dephosphorylation. So here what is happening? Either you're adding a phosphate group, that is phosphorylation, or removing a phosphate group, that is dephosphorylation. Now these phosphate groups, they control enzyme activity. The examples being glycogen phosphorylase. Then when it is phosphorylated, it becomes active. When it is dephosphorylated, it becomes inactive. Similarly, your glycogen synthase, when it is phosphorylated, that is, a phosphate group is added to glycogen synthase, then it becomes inactive. And when it is dephosphorylated, that is, phosphate group is removed from glycogen synthase, it becomes active. So basically, this is brought about by insulin and glucagon. So your insulin is going to dephosphorylate enzymes and then make them active. So all those enzymes which are under the effect of insulin, they are going to be active in their dephosphorylated state. And your glucagon, it is going to phosphorylate enzymes and then make them active. So all the enzymes under the influence of glucagon, they are going to be active when they are phosphorylated. So with this, we come to the end of regulation of enzymes. Once again, I'll briefly repeat it and then end the class. So I said, so enzyme regulation can either be brought about by controlling the quantity or controlling the quality, like I said. So when you're trying to control the quantity, what are you trying to control? You're trying to control the enzyme synthesis. And that is brought about by induction and repression. And this is taking place at the genetic level. Okay. So in induction, what is happening? You are using an inducer which is going to uplift or upgrade or increase or enhance the regulation of the structural genes that are going to synthesize that particular enzyme. So you're going to increase the amount of that enzyme synthesize. And in repression, what is happening? You're going to decrease the amount of that enzyme synthesize. For example, like I told you, ALA synthase. So when the ALA synthase is an enzyme which is involved in heme synthesis. When the level of the heme in the body is low, what will happen? The structural genes that are synthesizing your ALA synthase, they are going to be induced or activated. Okay. So what will happen? Your ALA synthase synthesis will be increased. More of ALA synthase will be produced, and hence what will happen? More of heme will be produced. So that is how you bring about enzyme regulation, by controlling the amount of enzymes synthesized. Then another way of regulating is through allosteric regulation. Now, what is allosteric regulation? It is taking place in allosteric enzymes, and allosteric enzymes are those enzymes in which the activity of the catalytic site depends upon the presence or absence of a modifier in another site, which is known as allosteric site. So in allosteric enzymes, what is happening? You have an active site, and apart from that active site, you also have an allosteric site. Now to this allosteric site, what is binding? Your modifiers are binding. Now these modifiers, they can either be activators or inhibitors. And to the catalytic site, your substrates are binding. Now, if the modifier that is binding to the allosteric site happens to be an activator, it is going to convert the unfavorable catalytic site into a favorable catalytic site, and your substrate will bind and enzyme will be activated. So that is allosteric activation. What is allosteric inhibition? In the allosteric inhibition, when the modifier, which happens to be an inhibitor, it binds to the allosteric site, what is happening? Your favorable catalytic site is going to change into an unfavorable catalytic site, and the substrate will not be able to bind to that site. And what will be happen? Your enzyme is inhibited or it becomes inactive. Now, another way of regulating enzymes is through covalent modification. Now, covalent modifications, there are again of two types, that is irreversible covalent modification and reversible covalent modification. In irreversible covalent modifications, you have zymogen activation. That is, your enzyme is initially produced in an inactive form, and then through proteolysis or breaking of covalent bonds, it is changed into an active form. And what is the significance of this? It is important because it protects the tissues from auto digestion and facilitates rapid activity. And examples are your proteases like your trypsinogen being converted into trypsin, your chymotrypsinogen being converted into chymotrypsin and all. So they're all examples of irreversible covalent modification. Then we have reversible covalent modification. What is that? That is you're adding or removing particular groups like somulation, ribosylation, phosphorylation, dephosphorylation. These are all examples of reversible covalent modification. For example, in phosphorylation, you're adding a phosphate group. In dephosphorylation, you're removing a phosphate group, and they are brought about basically by hormones insulin and glucagon. So insulin, it is going to dephosphorylate your key enzymes in the pathways and make them active, and your glucagon, it is going to make the key enzymes of the pathways active by phosphorylating them. So with this, we come to the end of today's class. We'll continue with what is left in the next class. I hope it was clear. I was a little quick because I was trying to cover up as much as we can. So any queries you can ask. Okay, see you then tomorrow. Thanks for watching.